Welcome everyone, my name is Todd Gaines and I'm a weed scientist at Colorado State University. Today I'm here to talk to you about biotechnology in wheat and applications in wheat breeding with special emphasis on gene editing, including CRISPR-Cas gene editing technology. What do we currently know about the status of biotechnology in plant breeding? Well, we certainly know that GM traits have been applied in a number of crops such as corn, soybean, and cotton, and these have been well accepted in a number of countries around the world and have been associated with more investment in those crops as well as yield gains, helping to improve the production for farmers in those systems. However, we also know that wheat as a crop does not have any GM traits, and we know that the yield growth for wheat needs to continue to increase. If you take a look at this graph, you can see that wheat production since the Green Revolution was increasing at a rate of around 2.5% per year. But since about 1990, that rate of increase has been just over 1% globally. We know, of course, we have a big challenge with a growing population to continue to produce more food, as well as protect the crops that we grow from the various pathogens, weeds, and insects that try to damage them. Breeders think about the genetic gain equation in terms of how quickly they can improve wheat yield. So this would involve things such as increasing the number of generations they can get in a cycle, how long that takes. They try to reduce the time. They try to improve their accuracy of selection. So they're doing a number of things around genetic gain. In wheat, one application of cell culture biotechnology that's getting some use is called doubled haploids. So this is a process where a breeder can make a cross between two inbred lines and then immediately fix those F1 progeny for heterozygosity and evaluate them for their improved yield potential. That's something that's been well applied in corn and now is getting more use in wheat, and that's exciting to see. Another application of biotechnology is genomic selection. This involves using high-density markers across the genome and understanding their predictive value for various traits such as yield, so that the breeder can actually make selections before putting lines into the field and spending a lot of money on breeding plots. So these are things that are helping to help breeders improve yield. But what about CRISPR? You would have heard, no doubt, in the news stories about CRISPR and gene editing and its huge potential. And so what is it exactly? You can see here on the cover of Nature, as well as Science, applications in animals and medicine. Various very high impact papers have come out reproducing mutations found in nature, and even applications of CRISPR to do things for human diseases. So it's very exciting, but what is it all about? Well, to understand CRISPR, we first need to think about what is a mutation. If you'll recall, the basis of genetics is DNA, and DNA consists of the four base pairs A, T, G, and C. You can think of these as being arranged kind of like the letters in the alphabet can make words and then sentences in a book. And you can move those letters around to make different words and make different sentences, but you know how to understand it because you know what those letters mean and you know what they mean in combination. That's essentially how the genetic code works as well. You have a combination of three letters that indicates a certain amino acid for the protein that's coded for by that gene. This genetic code is the same across all life forms. And then how this works as far as genetic diversity, how we can have different alleles of genes that underpin all the diversity that we see in life, is a mutation. So in this example, you can see part of a gene from wheat that normally has a G in the middle of this AGT, and that encodes a serine amino acid. But you can have a mutation from that G to an A, and then now that amino acid is an asparagine, abbreviated by N. Now keep in mind, the wheat genome is a hexaploid, so there are 3 full copies of the genome in wheat, totaling 16 billion base pairs of DNA. So if you think of DNA like a book, the wheat genome is kind of like a Russian novel. It's very long, but one mutation at one spot can have a big effect. And you might be familiar with this specific mutation because this is the basis of Clearfield wheat. So Clearfield wheat is resistant to the imidazolinone herbicides. If wheat has the serine at this position, it's going to be susceptible and it'll be controlled. If it has the asparagine, one mutation out of 16 billion base pairs, it'll be resistant. And these are the types of traits that breeders might work with to improve crops. Another mutation that you might be familiar with is in grapefruit. It turns out that the original grapefruit trees had fruit that was white and quite a bit more bitter, and it also had lower levels of a compound called lycopene. So naturally occurring mutations were found in which the fruit was more pink and a little bit more sweet. These have higher levels of lycopene, and then a really deep red grapefruit has even higher levels of lycopene. So mutations can naturally occur in populations. For the purpose of plant breeding, they could occur in related species, they could occur in maybe some distant populations or varieties from somewhere else, that kind of genetic diversity. We can also generate the same kind of mutations found in nature with a process called mutagenesis. This is when you would apply a chemical or radiation to seeds or plant tubers, other things, and you create random base pair changes, such as changing a C to a T. And these, in many cases, will have no effect. They could have a negative effect, but you do this to a lot of seeds, many hundreds of thousands, and you need a way to screen through them to find the one mutation that would be beneficial. So for example, in this case, some varieties of deep red grapefruit were created by a process of mutagenesis. So mutating the original white grapefruit and recovering the same type of mutation that also occurred naturally. Now, both of these are ongoing, and these are processes that are accepted to create plant products used around the world. This brings us to gene editing, because we can now create these same types of mutations, but in a targeted basis, and do them more quickly. So let's talk about how that works. So gene editing is a way to make very precise mutations in specific genes. There are multiple systems that have been discovered over the last number of years to do this, including zinc fingers, talons, and CRISPR-Cas, which is the one that has gotten special attention in the last few years. It's only a few years old, so keep in mind as we're talking about this, what was the status of computers 4 to 5 years after they were discovered, or smartphones. This is still very early stages of this technology. It's moving very quickly. So next we'll talk about what is CRISPR-Cas, how it was discovered, and how it can be applied in wheat breeding. The first thing you need to know is that this system was discovered in the bacteria E. coli. About 30 years ago, these sequences called CRISPR were discovered in the bacterial genome, which stands for clustered, regularly interspersed short palindromic repeats. When they were discovered, it wasn't really known what they were for, but just that this was a common pattern found in the genome of bacteria. This is a good example of really basic research later on leading to some big applications. So in 2002, these genes called Cas, which stands for CRISPR-associated, were found, and this is a protein that can cut DNA like scissors. And these types of genes were also found in the genome of bacteria. Now, this brings us to the question of the bacterial immune system. We know that in animals, there's a protein-based recognition. So if an animal is exposed to a pathogen, the immune system can develop antibodies that recognize certain protein patterns in that pathogen and use that to trigger an immune response the next time that pathogen is encountered. So bacteria also have pathogens, but they have viruses that cause diseases in the bacterial cell. So this is a nucleic acid-based immune system for bacteria involving the CRISPR-Cas. So let's take a look at how this works. In 2005, this was discovered that CRISPR and Cas are a bacterial defense against viruses. So how does this work? The bacteria has these Cas genes in its genome, these molecular scissors, and it will encounter at some point a virus that it hasn't encountered before. The virus will inject the viral DNA, and that's going to cause whatever infection pathogenesis, but the bacteria will capture a piece of that DNA and actually incorporate it in the genome to retain it for the future. And this will happen repeatedly. You might find several different markers from previous infections in a bacterial genome. So now when the progeny of that bacteria encounter the virus again, that viral DNA goes into the cell, the nuclease will be activated, the Cas molecular scissors, along with a guide that is transcribed from the signal of the virus in the bacterial genome. Now, this guide is going to go and precisely match the viral DNA in the cell, and once that match happens, the scissors of the nuclease will cut up the viral DNA and destroy it and protect the bacteria from the infection. So therefore, now the application of this is a guided delivery system for modifying DNA and making an exact mutation. So you have in this the component of the molecular scissors, the Cas gene, and the guide. So in the bacteria, this guide comes from the genome. So that guide is going to pair up with the molecular scissors, and the sequence of the virus DNA will look something like this. And what's really critical here is you'll have an exact match between the guide that's transcribed from the genome and the virus. And when these come together, the nuclease will cut it, and that destroys the DNA to inactivate it. So then all of this had been discovered, and the idea then came up, okay, you could change and swap out that guide for anything. Could you do this in other types of cells besides bacteria? Could you do this in animal cells and plant cells? So I'll show you an example. We know in a lot of cereals, such as wheat and barley, there's a disease called powdery mildew that can cause severe infections and yield loss. In barley, there's a gene called MLL1 that's been studied for some time that can cause resistance to powdery mildew. Normally, this gene is active, and it actually functions to suppress the defense mechanisms against powdery mildew, and the barley plant will be susceptible. However, in a resistant barley line, this gene is knocked out. This is a naturally occurring mutation, and this allows the defense mechanisms to be activated, and it makes the plant resistant to powdery mildew. So how could this be applied then in wheat? Wheat, of course, is a hexaploid, and each of the genomes has a copy of MLL1. So there were no known sources of mutations in all three genomes in wheat. However, then, you can take the system and make a guide, this piece right here, that would actually match the wheat MLL1 gene and target the copy on all three genomes, so that this can be inserted into the wheat cell, and the guide will match the DNA, the nuclease will then cut. And what happens next? Well, now you have the MLL1 gene with a cut in it, and there are repair mechanisms in cells whenever these types of cuts are found to fix them. But sometimes it's fixed with an error, and this is normal. It doesn't happen all the time, but it might happen at something like 5%. So if you do enough cell lines, you'll get sometimes an error inserted, and that might change the coding structure of the gene so that it doesn't work. Or it might even result in part of the DNA being deleted so that it's not there anymore. And then in wheat, what this resulted in, again, you have susceptible wheat, 3 copies of the MLL1 gene. You can see that it's infected by powdery mildew. However, in this genome-edited wheat line, all 3 copies of MLL1 were knocked out, and this allows the defense mechanisms to be activated, and now the plant is resistant. So this was published in 2014, really exciting progress to show that this can be applied in wheat. Here's an example of something else that might be done. Let's say you want to make a specific change, not just knock out a gene, but you want to change a base pair to change the amino acid of a protein. What might that look like? Well, in this example, we show hypothetically, let's say if you have a G at a position in this gene, you have a normal size wheat grain. But if you were to change it to a C, you would have 5% larger wheat grains, as an example. How would you do this? How would you change a G to a C? Well, again, you would design a guide that would go and match that specific gene that you're interested in, along with the Cas protein, the scissors. So the guide will go and bring in the nuclease and cause a cut right at that point. Then now you have an open strand of DNA. What's going to happen? You need another component. You also express in the cell the DNA template with the change that you wish to make. So this is what's called oligonucleotide mediated repair, or introducing a piece of DNA that the plant will incorporate to make the change that you want. So in this case, you can see it has the C, and in the cells in which DNA repair happens with the alternative template, now you'll have a C at that position instead of a G. And then the cells and the plants that contain that mutation will have the larger grains. How's this then being applied in other areas in plant breeding? Well, there's a trait called waxy corn, and this is a CRISPR trait that's been developed by the company Pioneer. Waxy corn has a different starch profile. It has some industrial uses, and there's a naturally occurring mutation for it. So this is an existing product on the market. However, the original source from where the waxy corn was identified has what's called a linkage drag. So it has lower yield because of other things that are closely linked to this waxy gene. Now that this can be done with CRISPR, that only that exact change is made, nothing else, now you can have waxy corn without any yield drag. And Pioneer currently has this trait submitted for regulatory review. So we'll see where that goes. What are other types of things that can be done in wheat? I think it's important to know there is a lot of potential. You couldn't just do anything. The key thing is you need to understand what gene it is that you want to make a mutation in, and those are where applications are possible. One example that's been published is taking a look at the gluten in wheat, which is a complex of protein and other components, but there are certain parts of the protein that we know are the components that trigger celiac. And that has been found in the wheat genome to have about 45 copies of these types of genes. So a group has knocked out 35 of those. This could potentially result in a wheat flour that contains gluten with all its elastic properties, but that wouldn't trigger celiac. So then you could have, in essence, something to help address what currently, people who need to be gluten-free might then be able to consume wheat flour again if those protein components were knocked out. It can really facilitate faster transfer across varieties. For example, let's say a disease resistance gene is discovered in Europe and Australian wheat breeders wish to cross with it. Well, rather than having to import the seed, wait for quarantine, do a seed bulk up, do crossing, etc., a number of years, they could go in and make that mutation in their adapted varieties and get there much faster. Herbicide resistance traits could be introduced into wheat with the known basis of herbicide resistance for various things that might be of interest. Drought tolerance genes, as more information is discovered from basic science about how drought tolerance can be controlled. Those are types of traits that could go into wheat. And then quality is a big one. For example, we know that for some types of noodles, it's important that the wheat flour doesn't turn brown once it's mixed with water and exposed to air. There's a gene called polyphenol oxidase that's in plant cells that causes that browning. Think of when you cut into an apple or a mushroom, and with a bit of time in the air, they start to turn brown. We can knock out some of those genes in wheat and reduce the browning and have a better quality product, or perhaps look at things like protein. And then disease resistance as well has some potential for applications with gene editing. What about the regulatory process? Well, this is a big question because are plants produced using CRISPR GM plants for the purpose of regulation, for the purpose of public opinion? I would say there's a general scientific consensus that single base pair mutations can occur naturally. We can generate them more quickly through mutagenesis, and now we can make them quite precisely with gene editing. All of these would, in the end, produce the same product, and therefore I think there's a consensus that the regulation of CRISPR should be similar to how mutagenesis is regulated around the world, and that there's a general acceptance for products derived from mutagenesis and plant breeding. Again, I think it's very important to think about what that product is, but in terms of the process itself, gene editing has a lot of great features to make it a safe process to use in plant breeding. We do know that off-target effects can occur of CRISPR gene editing. The technology is really moving quickly. Better versions of the Cas protein are being discovered that have fewer off-target effects. It's possible to kind of inject the guide RNA and the protein into a cell without having to actually insert those genes into the genome to make it happen. That has less off-target effects. So that's an area that I think will continue to improve. Certainly, regulatory agencies around the world are currently deliberating this, and we'll need to wait and see what their decisions will be regarding products produced using CRISPR. So to summarize, again, what is gene editing? With the CRISPR-Cas system, it's a molecular scissors. This photo is maybe a bit hard to see on this projection, but this really bright spot is the Cas protein, and it's attached to a DNA strand. And right here, there's a loop of DNA. It's been captured. It's just made the cut. So you can really see, and this is a screen capture of a video from a scientist from Japan, and it's just quite incredible to see this protein cut the DNA. Gene editing mimics naturally occurring mutations. What this can do is reduce the time for breeding cycles and help get products into the field faster. From basic biology perspective, it allows us to study the genetic basis of traits, which can also then result in better products for growers and consumers. There are technical challenges in wheat, especially the hexaploid nature and the cell culture aspects of wheat, but that area is progressing quite quickly as well. There will most likely be some costs associated with products produced using CRISPR-Cas. Right now, Pioneer has the licenses from both Caribou and the Broad Institute for IP uses of CRISPR gene editing in agriculture. But certainly there's a philosophy here that they want to see this technology broadly applied. And it's free to use for researchers, and someone commercializing a product from this will need to negotiate a license agreement. But I think it's going to be set up in a way to make it very accessible and really encourage people to use this and to get products out there. So with that, I'd like to thank the Colorado Wheat Research Foundation for their support of our work at Colorado State. I'd like to thank the Australian Herbicide Resistance Initiative for all of their input into the weed science research. And I'd also like to thank my colleague, Dr. Stephen Pierce at Colorado State University for his assistance with the slides illustrating the CRISPR-Cas gene editing. And thanks very much.